Hello, this is Mr. McMurray, and it's about 10 o'clock at night, and yep, sorry a little bit late today, but computer issues have kind of pushed us on a little farther. But here we are. We are ready to go, and this is the protein synthesis drawing. All right, so we're going to go over this, discuss it, and then tomorrow we will have notes that go along with it. But for now, just worry about the drawing and getting it labeled. All right, so starting off, if we're going to make protein synthesis, we are going to start with a process known as translation. And it's going to occur inside the nuclear membrane. You notice here's a nuclear membrane. This is the nucleus. This would be out in the cytoplasm of the cell. And if you remember, the DNA, which is the instructions for building proteins, are here inside the nucleus. The DNA cannot leave the nucleus because if it did, it might get damaged. If it gets damaged, the cell might die. And that's a bad thing for the cell, and since you're made out of cells, that could be a bad thing for you. So instead, it is going to stay inside here and make a copy of itself. And that's the first step. It's called transcription. All right, transcription, to transcribe, means to copy across from something else. In this case, we have DNA strands, and we're going to have messenger RNA nucleotides lined up on one side of it, and make a copy, except for it's going to be an RNA copy of the actual DNA. All right. So to label a few things here, number 14 here, this is the DNA strand that has unzipped here in the middle so that it can, the RNA, messenger RNA can come in and line up there. These other things floating around here then represent nucleotides, in particular RNA nucleotides. Number 10 down here would be an RNA nucleotide. And a nucleotide we know is made up of three parts. The first little cross bar here that matches up right across are the nitrogen bases. I put an N for nitrogen, but nitrogen bases, and they are attached to a square here right there. Okay, and that is the sugar. Now specifically, if this is an RNA nucleotide, we know the sugar would be ribose. That is correct. If it were DNA, sugar over here in DNA, that would be deoxyribose. But anyhow, that's the sugar. And then the sugars are attached to the next sugar in the chain. As you can see here, you got a sugar and there's a little dot between it. That is called the phosphate group, or PO4 group in this case, because PO4 is the chemical formula for phosphate. All right, and when you hook them all together, they make this strand of messenger RNA, which is number 9, messenger RNA, or little m RNA. So notice it's made out of RNA here. And this is an exact copy of one side over here. So, for example, if you had A here, you would have a U lining up across from it because A pairs up with U in RNA. There is no T in RNA. If this was a G, that would be a C. If that was a C, that would be a G, and so on. And so it would have paired up and matched that. So it's a copy, although it doesn't have the same bases. It has the opposite, or the complementary base that goes with it. All right. Now, it has to leave the nucleus, so it goes through the nuclear pore. You can tell it's not the nuclear rich because they would have a gate here, and it'd be some kind of gated community or something, probably with guards, no less. All right, but anyhow, so it can just slip right out here and go out into the cytoplasm. Now, we have the instructions. Now we need some place to build it. Well, the place we build proteins, if we remember, are ribosomes. This is a little mushroom shaped here, another one here, and another one up here. These are all ribosomes. So now we're going to take the instructions to the ribosome and we're going to build a protein. So this is kind of like going from a bicycle instructions and a box of parts to actually putting it together to make a bicycle. All right? And this part is called translation. So we're going to translate the instructions into an actual protein chain, which is what this represents up here. Okay, now to do that, we have to move the messenger RNA to the ribosome. So we got the instructions to the factory. But we can't build anything unless we have the right parts. And proteins tend to be built out of amino acids, which are what these little round things are floating around here with names in them. You can't read them very well, but those are names of various amino acids. But we need to get the right amino acid over here to the ribosome. So we are going to, first of all, label ribosomes. Remember, they are made out of rRNA, or ribosomal RNA. And so they are actually another type of RNA. It's going to be all in the family here because we have messenger RNA and we have rRNA. All right, and there's going to be another RNA in a minute. We'll talk about tRNA, but amino acids, that's what we need to get to the ribosome, and we're going to use tRNA. T stands for transfer, because we're transferring the amino acids from floating around out here in the cytoplasm into the ribosome where they can be used to make a protein. If you want to think of it as a truck, these would be pick-em-up trucks, because they're going to go pick them up a amino acid and bring it back to the ribosome. You can see one coming in here, one that's already come in here, right there. All right. Now, when they bring the stuff, people have been known to go to the store and make mistakes, especially people who drive pickup trucks. So it's possible they got the wrong thing. So we need to find a way to check and make sure that we have the right one. The way we do that is every 3 letters on the messenger RNA, like here's G, U, A, and here's the last here, is a codon. That's like when it's cold outside, you need to make sure you put a codon. At least that's what your mom told you to do. Anyhow, that's not the kind of codon it is. Okay, this is a code. It's a code for one of these amino acids. All right. There's another code on the tRNA. When it comes in with the amino acid, it comes in here, and it has 3 bases that line up on the outside. Now you can see this is just a strand of RNA that's been twisted around into kind of a clover leaf or cross shape. On the inside, there would be little bases in here, and that's what helped bend it and make it into the shape. But on the bottom here, there's 3 that face toward the outside. And those 3 are called the anti-codon. All right? This is like the kid who doesn't want to put his coat on when he goes outside. Just kidding, not really. But anti-codon in this case, because they line up against the codon, and they see if they match up. Now, if they match up, G and C, U and A, U and A again over here, okay? Then if they match up and it is the right one, then that amino acid is hooked on to these other amino acids, which makes a protein chain. Or sometimes these are also called polypeptide chains. Poly means many, peptide just means has to do with proteins. And so sometimes we talk about peptide bonds that hold these together. So yeah, this is a polypeptide or a protein chain. Now this chain only has about 4 on it. Eventually we'll have anywhere from 100 to 1,000 of these amino acids in order to make the complete protein. All right, so this would just keep going. The messenger RNA would scoot down 3. The next tRNA would come in. Hopefully it would match up with those next 3. If it did, then its amino acid would be added on. And it's just going to continue to do that very quickly until it has all the proteins hooked together and make it. Now, obviously, it goes a lot faster than what we're talking about. It goes so fast, boom, 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 they're just bringing them in, boom, boom, boom. And it's amazing that your cells are doing this all the time, constantly building proteins to be used for enzymes, to make the different reactions in your body happen, speed up, slow down, whatever. And they do this so quickly that they can build these 100 to 1,000 amino acid in a minute or less. Okay, they can do this very quickly. So that is pretty amazing. All right. Other than that, that's all for the drawing. Tomorrow we will take some notes on the process, but basically it's going to be the same kind of process that we talked about here. And so anyhow, we'll do that. So if you're too late for you to get this done tonight, just get it tomorrow. I'll make it due tomorrow. And then you can also do the notes. All right. Otherwise, have a good night, and we'll see you tomorrow.